The topic of work is in vivo enhancement of anticancer therapy using bio or chemotherapeutic drug-bearing nanodiamond particles. Next, I introduce our work. First, the effect of pH and ND-DOX adsorption and release by ND was studied. It shows that in the sodium hydroxide solution of pH 10, ND had a great test adsorption capacity for DOX. The slow DOX release rate observed at pH 7.4 for ND-DOX nanoparticles. Second, TM and FTIR were used to verify the formation of ND-DOX complex. Third, as the figure shows, it implied that the most amount of DOX is released from ND-DOX around the nucleus. These results showed ND-DOX is capable of mediating controlled drug release and has a slower and more sustained drug release profile. Then, tumor-bearing mice were treated weekly with ND-DOX, ND or PBS. Strikingly, we found that ND treatment significantly can effectively inhibit tumor growth. In addition, an interesting phenomenon was found that the survival rate of tumor-bearing mice for ND-DOX nanoparticle treatment is 4 times that of DOX treatment. More exciting, survival rate for ND-treated mice is close to that of DOX treated. The result is a novel to our knowledge. It is a schematic diagram for ND or ND-DOX nanoparticle transport in normal and tumor tissue. Finally, histopathological analysis displayed that ND and ND-DOX have no apparent side effects as various tissue, while the toxic effects of DOX are obvious. In summer, ND can be a potential material of prolonged survival lifespan applications, and ND-DOX can act as a nanodrug with a promise enhancing chemotherapy efficacy and safety. Future perspective: one is the distribution of ND or ND-DOX in animals. The second is the excretion of ND nanoparticles is of great interest and importance after DOX release from ND-DOX nanomedicine in vivo. That's all. Thank you.